Alright guys, welcome back to PS Pain Relief. This is week 7 and we'll be going over muscles, blood, pharm, and cancer. So Christina and I will be going over muscles and vasculature first. I'll be taking you through the different types of muscles, and then Christina will take you through histology, muscle organization, their activation, and the histology of our different blood vessels. Right, so starting off with the purpose of muscles. So the most obvious one that we all know is just movement, but the other two we might not think of as much. And the first is just temperature regulation. So depending on the temperature, they can control the blood flow to the skin, and they can also allow us to shiver, which produces heat. And from our last topic, from metabolism, we'll know that it's also the body's biggest source of protein and amino acids. So some features of muscles are that they contract, and they're elastic. So the muscle fibers can stretch beyond their usual length and then bounce back. And lastly is they're capable of hypertrophy and hyperplasia. So they're able to increase in size and also increase in number. So the components of a muscle cell are myofibrils. So these are the microfilaments actin and myosin, and they're basically cytoskeleton. There's a sarcoplasm, which is just a muscle form of cytoplasm, and the sarcolemma, which is the muscle cell membrane, and there's sarcomere. So they're the smallest repetitive contractile subunit. So yeah, they're just like the smallest, I guess, building blocks of muscles. I guess you can say that. They're found only in skeletal cardiac muscles, and they range from Z line to Z line. So if you look here, this is a sarcomere from one Z line to another Z line. Yeah, and there's the sarcoplasmic reticulum. So it is the muscle's equivalent of a smooth ER, and it stores calcium that is used to activate muscles, which you'll see later. And lastly, there's a neuromuscular junction. So it's not shown in this diagram, but it's just basically where a neuron kind of meets a muscle and where it activates the muscle. Oh. Okay, we're gonna start with skeletal muscles. So they're cylindrical, and they have multiple nuclei that are found in the periphery. They have a lot of vascular supplies, and they have striations. And lastly, they have these things called T-tubules like this. So they're just extensions of like the cell membrane, the sarcolemma, that go down to the cell. And so this way, when it depolarizes, all the ions can flow all the way down here and depolarize the whole muscle, because otherwise the action potential will only work on the periphery, only on the outside, but this allows it to come down and come in. And these are, they have complexes called triads. So each T-tubule has basically like two terminal cisternae. That's how you pronounce it, on either side. Yeah, so it's just like a triad because it's got 3 components to it, like 1, 2, 3. So there's a sliding filament theory, and it's the theory of how muscles contract. And they're basically, it basically means that the muscles contract by the interaction between thick and thin filaments. So thick myosin and thin actin filaments, which you can see here. So myosin filaments are thick, and they have heads. They have heads that interact with actin filaments. Yeah, and actin are just thinner, like smaller filaments. And so in a sarcomere, you'll see A bands and you'll see I bands. So A bands, how do I move this way? A bands are anisotropic, meaning that they don't move, they don't change, and they're broad and dark because that's where thick and thin filaments overlap. But I bands are isotropic because they move and then they disappear when like muscles contract, or they like shorten. Yep. And so muscles contract using calcium and ATP, and sarcomeres shorten during contraction. But just because they shorten doesn't mean the individual filaments get any smaller. They don't change in length. All they do is overlap over each other. So yeah, when a muscle contracts, Z lines move closer to each other and M lines move closer to Z lines, and the H band disappears. So yeah, it's kind of complicated to be able to identify what each band is, what each line is, and I recommend you guys just go over it in your own time, draw it a few times yourself to just remember this. And I'm not going to go into it any further because I'm pretty sure Christina covers it in more detail later. Right, okay. So the development of skeletal muscles involves, so myoblasts is like, I guess you could call it like the precursor of a myocyte, so like a muscle cell. So they fuse and to elongate the fiber. So instead of one myoblast growing to be really long into a muscle fiber, that's not how it works. It's multiple cells that fuse together to become a fiber. That's why myocytes have multiple nucleuses, because they're actually made of like a lot of different cells. And when they're like first, I guess like embryologically, they're from like different cells, and they differentiate. And as they differentiate, they begin to produce myosin. And there are different types of muscle fibers. This is really low yield, so you don't really need to know this at all. Basically, there's type 1 slow twitch fibers and type 2 fast twitch fibers. And type 1 fibers is smaller than type 2. It's easier if you just look at this diagram. So type 1 slow twitch ones are more for endurance, long distance, while type 2A is kind of the intermediate, and type 2B is for like short bursts of energy, I guess. Yeah, so the fatigue rate of type 1 is low because they have greater endurance, and the fatigue rate of type 2B is greater because they just produce a lot of energy at once. So this is more detail in it. So slow twitch fibers have many mitochondria and myoglobin. So yeah, myoglobin is used to store oxygen. They have greatest resistance to fatigue, but generates less tensional power. And type 2A are known as fast twitch oxidative fibers. They have many mitochondria, many myoglobin, and a lot of glycogen. And they use a lot of anaerobic glycolysis. Yeah, and lastly, type 2B has less of the first two, less of mitochondria and myoglobin, but stores a lot of glycogen. And they fatigue rapidly due to lactic acid production, and they contract rapidly. So they're used to precise, fine movements and short bursts of energy. Yeah, now we're gonna move on to cardiac muscles. Okay, so in development, the cells join together at junctions. And as the heart increases in size with development, cardiomyocytes disassemble their contractile filaments to undergo meiosis. Okay, that's, yep. Yeah, and they don't grow much postnatally as they have limited regenerative capacity. I guess we'll see all this later, but this is just on the side. You'll see that they join with this. Yeah, because that's it. And for the structure, they're also striated, but they're branched. And the reason they're branched is, you'll learn this later, but basically the heart is a huge piece of like, it's made of muscles, and you want your heart to contract at once to beat and like push your blood around. That's why by being branched, it allows the depolarization to spread between cells much faster, and they have one or two central nuclei. So skeletal muscles are multinucleated and on the periphery, but for cardiacs, there are one or two in the center. Yeah. They also have sarcomeres similar to skeletal muscles with A and I bands. And instead of triads, they have dyads. So it's one T tubule with a sarcoplastic reticulum next to it, like this. So in a skeletal muscle, remember, it was a T tubule with the sarcoplastic reticulum on both sides, but with cardiac muscles, it's only on one side, so it's a dyad because there's only two. And they have a lot of mitochondria for high aerobic respiration, because of course the heart can never stop beating unless you're dead. And they've intercalated this. So they're, if you remember from, what was it histology? Yeah, you learn how different cells are joined. So they have adherents and desmosomes, which allows the cardiomyocytes to adhere together to stick together. So basically the heart doesn't fall apart. And then they have gap junctions. So they're like pores in the cell that allows for connection between cytoplasms of adjacent cells. And this allows ions to flow through them, but not other things. So only ions can flow through them. And this allows the heart to contract simultaneously. Yeah, and lastly, we're going to move on to smooth muscles. So unlike cardiac muscles and skeletal muscles, they have no striations, but they still rely on actin-myosin interaction and calcium for contraction. And so yeah, they're spindle-shaped cells. They're stacked together and in a lattice-like structure instead of sarcomeres. They have one nucleus and they're located in the center. They have actin-binding dense plaques in the sarcoplasm, which are equivalent of Z-lines and striated muscles. and they contract slower but holds the contraction for much longer. And the reason they contract slower is because they have no tubules. And the reason that they need to contract, or the reason they don't need to contract fast, is because so smooth muscles are found in all of these like kind of structures. So basically, they're like found in hollow tubes or organs such as the esophagus, the bladder, stuff like that, and or like especially like the small intestines, the big intestines, and all of these structures don't need to contract at once. Instead, they mostly move like in peristalsis, such as in the small or like small or big bowel. So they're just trying to move food along. So you just contract the piece and the next piece down and the next piece afterwards. So there's no reason for the whole muscle to contract at once. So that's why they don't have to contract faster. Yeah, and they have less mitochondria as a result. And the muscles, well, the organs that they innervate are the ones, not innervate, the organs they're found in are the ones innervated by the autonomic nervous system. So these are all like examples of it. And lastly is the repair and regenerative ability of well, these different type of muscle cells. So smooth muscle cells come first, and it's skeletal, and then it's cardiac muscles. So this is like how well they can repair themselves when they're damaged. And smooth muscles, they're highly versatile and can change their phenotype. So this means that they can continue growing, they're proliferative, and they can continue to differentiate and stuff. So examples are such as like a growing uterus in a pregnant person, or like the wound healing of blood vessels, because smooth muscles are found in blood vessels. So all of these stuff, they can grow, or if they break, they can be healed. And of course, they can undergo hyperplasia and hypertrophy. On the other hand, skeletal muscles, they only contain satellite cells, which is found under the basal lamina. They're stimulated to divide when the muscle is damaged, and they fuse with existing fibers. And, however, cardiac cells don't have satellite cells at all. Thus, they're basically irreplaceable once they die. So it's like when you have a heart attack. I'm not sure if you guys have covered this yet, but when you have a heart attack and pieces of your muscle cells die, they can never come back again. Instead, they're replaced with collagen. They're not replaced with more cardiac cells. So yeah, basically the reason for this is skeletal and cardiac muscles undergo terminal differentiation. So in adults, the organs retain their phenotype. So basically, when they're first embryological cells, after they are differentiated, that's it for them. They can't differentiate anymore. They can't proliferate anymore. Yeah, so the original muscle fibers can't divide anymore, but they can hypertrophy instead by having, for example, in skeletal cells, by having these satellite cells to lay down new protein and enlarge them. But the original fibers are still the same. There are no new ones. Yeah, and that's it for me.
But to start us off, the three main muscle types we'll be talking about are skeletal, cardiac, and smooth muscle. So we've divided it based on their features, neural control, and connections. Both skeletal and cardiac muscle are striated, which means they've just got lines through them. Skeletal muscle has multiple peripheral nuclei, so they have more than one nucleus, and they're found in the periphery, so along the borders of the cells. They're also under somatic or voluntary control, so you can decide if you want to contract a skeletal muscle or not. And they're connected to bones via tendons, connected via tendons and fascicle bundles. We move on to the cardiac muscle. So this cardiac muscle is found in the heart. It's also striated, and it has intercalated discs. The fibers are also branched, and you can see central nucleus. They're also under involuntary nerve input, so you can't decide how much or how hard you're going to pump your blood. You can't decide that. And the connections between cells are via end-to-end cell junctions. And you guys can see this photo over the next page. And last of all, you got that smooth muscle. No striation here. They're spindle shaped, so they're quite cylindrical shaped. And involuntary as well, so you can't decide how your smooth muscle contracts and works. And the connections are via connective tissue with gap and desmosome junctions. But that's enough with this table. I'd much rather show you guys a photo. So here we've got some histology just up top, what it looks like under a scan, and some annotated of just some images. So here, skeletal muscle, you can see the peripheral nuclei, they're mostly along the edges. You can also see the striations, the stripes, right? A muscle fiber, so you can see the length of a muscle fiber. You can just see from that like light zone, dark zone, just in like a rectangular shape. And there's some connective tissue connecting the skeletal muscle, and that's again what it looks like over here. In the middle, we've got cardiac muscle. You can see those intercalated discs here, the connections right here. You've got those centrally located nuclei, and you can see the branching as well. So it's not just straight linear like skeletal, it branches. And yeah, there'll be glycogen and whatnot in between these spaces. And then last of all, that smooth muscle. It's not as linear, not striated. They're cylindrical, and you've got the nuclei. So don't really have a direction. Over the next couple of slides, I've got the same thing, histological images of both skeletal, cardiac, and smooth muscle. And you can kind of see they're annotated as well. So pay attention to the fact that it's striated. You can kind of see these stripes running throughout. There'll be like a fiber. There's multiple peripheral nuclei, so found along the edges, right along here. Somatic control, tendons and fascicle bundles, like we've kind of mentioned earlier. Moving on, cardiac muscle, it's kind of different. So again, you notice those striations. You should be able to see there's the striping along here, those vertical striping. You'd also notice, however, some significant intercalated discs. Now, what these are is the connections. You've got branching fibers. So if I just clear the drawings here and follow some of these fibers, you've got this one going down to the bottom, this one going down to the bottom. There's another one branching up top here. So you've got that branching fiber. And another thing which could help you distinguish cardiac, so heart muscle, from skeletal muscle are those centrally located nuclei. So you don't have multiple peripheral nuclei. Reminder, it's involuntary. There's end-to-end junctions between cells. And yeah, that's cardiac muscle for you, which means our last one, and you guys can always go back, have a look at these images again later. I think they'll be covered in your active learning. Last of all, smooth muscle. No striations. It's not as organized. And here you can kind of see the spindle-shaped cell. The fact that it looks a bit like this weird flattened cylinder doesn't really have much direction. They're kind of stacked on top of each other. And that's what you'd notice in your smooth muscle histology. So those are the three types of muscles. Now, let's focus a little bit more on skeletal muscle, and we'll be talking you guys through muscle organization. So starting from big, going to small, you've got a big skeletal muscle, just right here, big skeletal muscle, right? Now, that is split into what we call a muscle fascicle. So one of these little groups is called a muscle fascicle. We zoom in from there. From a muscle fascicle, we can see that these are composed out of muscle fibers. Now, I can tell you, a muscle fiber is essentially just a cell. You know, a muscle cell is the same thing as a muscle fiber. If you have a look at these diagrams, you'll also notice that the skeletal muscle is wrapped by a layer of epimysium. The muscle fascicles are wrapped around by a layer of perimysium. So this is just a connective tissue around it. And you've got endomysium covering the muscle fibers. So from big to small, you've got epi, peri, and endomysium. And what these contain is ECM, so extracellular matrix. They carry nutrients to the muscles and whatnot. If we have a look at a histo image here, this shows you a muscle fascicle. So how do we know it's a muscle fascicle? We can see a bit of endomysium, which is between the muscle fibers. So each of these little units is a muscle fiber, an individual muscle cell. And you can see how they've zoomed in here, and you can see those individual muscle myofibrils, all those little dots, those are myofibril. Within this muscle fascicle, you'll also see them peripherally placed nuclei, and you see multiple of them. Whereas if we were just looking at just the muscle fiber, we wouldn't see this many peripheral nuclei. We'd be looking at just one of these zoomed up really close. And if we were looking at skeletal muscle, we wouldn't be able to see those slight little distinctions in the myofibrils. Okay. So again, have a look at these images again later. Just really have a look at all the labels and what's going on in there. But that's muscle organization for you. From big to small, you've got skeletal muscle, muscle fascicles, and those muscle fibers or muscle cells. Each muscle fiber will have many myofibrils within them. And that's what we're going to be talking about when we get into muscle activation. How do we send a signal causing a muscle to contract? So muscle activation, again, Starting out from big, you've got your skeletal muscle here. So say, for example, your bicep. You've got the tendon connecting it to the bone here. We zoom into the muscle, we have a look, right? And we can see the muscle bundle. We look at, from muscle bundle, you've got the muscle fascicles. Within each muscle fascicles, there are muscle fibers. And then within each muscle fiber or each muscle cell, there'll be many myofibrils. So again, muscle fiber, muscle fibril. And it's within the unit of the muscle myofibril that we're going to be talking about things like Z-line, actin, myosin, sarcomere, a whole lot going on. Oh, let me just erase this. So first of all, couple terms. First of all, we want to know what a sarcomere is. Sarcomere, you see right here, is the functional unit of a skeletal muscle fiber. So this is the functional unit which is able to contract, able to shorten. What a sarcomere has is proteins, both actin and myosin filaments. So actin are the thin filaments, myosin are the thick filaments. So if we just have a look down here, you'll see actin are thin green filaments and myosin are thick purple filaments. Now, actin and myosin have a role in muscle contraction and how things work. We'll go into that in a little bit more detail over the next slide, but there's a couple things you guys need to know in terms of muscle activation. However, before we move on, sarcomeres. So sarcomeres are basically found within myofibrils. So we're talking muscle fiber, muscle cell has many myofibrils. Now, myofibrils will have sarcomeres, which is a small contractile unit. Now, hundreds and thousands of myofibrils have to contract before a muscle is able to contract. So we can kind of think about the level of contraction here. All these small little guys will need to contract so that one muscle can contract. So that's what a sarcomere is, the functional unit. And you've got the sarcoplasm. Sarcoplasm is basically just a special type of smooth ER. Its role is pretty much to do with calcium, iron storage, release, and retrieval, and that's to do with muscle contraction, the nerve impulse arriving. But before we go and have a look at all that fun stuff, it'll be helpful to just talk about this sarcomere and talking about things we notice here. So first of all, identifying the actin and myosin. So it's green down here. Up on this image on the left-hand side, it's these thin little bands right here. These are your actin filaments. Your myosin are these like red, thicker ones in the middle. So that's your myosin. Okay, so when we have a look at this diagram, we've got a couple of things to talk about. First of all, the Z-line. What is the Z-line? And you guys will be able to find these in the slide notes as well, just underneath. The Z-line is the anchor point for an actin filament. So this is where the actin filaments anchor along this Z-line. You can also see it right here, the green actin filament anchor point, okay? Now, a sarcomere starts and ends at the Z-line, which means a sarcomere is this length from this Z-line all the way to this next Z-line right here. That's the length of the sarcomere. Now, M, I think, is nice and straightforward. The M line is like the middle. In here, you find only myosin filament, okay? So it'll be really important just to line is Z-line, H, A, and I. I think it's like one or two multiple choice questions on the exam. But regardless, it's just helpful to know so you're not completely confused during the class. So again, Z-line, anchor point for the actin filaments, only actin. M-line is the middle, and that's where you find myosin. So when I hear M, I think middle, and I think myosin only. Okay, from there, let me just clear this little space. We also need to talk about the light I band. So this light I band only contains actin. And then you've got the dark A band, which only contains myosin as well. Okay, and H zone contains, but H zone is the area between, where it's just myosin as well, between the two segments where there is actin, okay? So this is a sarcomere, this is one unit. Many sarcomeres in a myofibril, many myofibrils in a muscle fiber or a single muscle cell, and muscle cell to muscle fascicle to bundle or skeletal muscle. So I hope you guys are getting the idea of the layers here. And also gaining familiarity with the sarcomere, the small contractile unit of muscle, and sarcoplasm, a special type of smooth ER responsible in calcium storage, release, and removal. Okay, So with that in mind, having talked about a sarcomere and what it looks like, let's move on and try to talk about how muscles contract and the kind of process involved. So try bear with me, guys. So recap, where were we? We're at the neuromuscular junction, so the junction between the neuron and the muscle, where the muscle and the neuron meet. You've got an action potential, so that electrical message traveling along the axon terminal, so insulated by these myelin sheaths, they get to the axon terminal, and that's where it arrives at the neuromuscular junction. As usual, you've got your neurotransmitters, acetylcholine, being released, diffuse across the neuromuscular junction, and it will bind to receptors. When it binds to those receptors, it will open sodium ion channels, and basically it will cause a new action potential in the sarcolemma. We talk about the sarcolemma, so sarcoplasmic reticulum. It was the one involved in calcium storage, release, and retrieval, right? So you've basically sent an action potential down, diffusion, and you get a new sodium channels open, you get a new action potential traveling along the sarcoplasm. And the sarcoplasm and sarcolemma, same thing. So sarcoplasmic reticulum and sarcolemma are the same thing, and they're distributing this action potential or this impulse along the length of a muscle, so that we target not just one single muscle myofibril, but we're getting a lot of them. We're spreading this message, right? So the action potential travels along T-tubules, that's just what they're called, travels along these T-tubules, and it depolarizes the membrane, including T-tubules. And what happens here is the sarcoplasmic reticulum will release calcium, okay? It gets a bit more complicated in a little bit, but I'll just try to explain it through the rest of this diagram. Calcium is released from the sarcoplasmic reticulum. What it does is it binds to a protein called troponin, causes actin to become unshielded, and then cross bridges can form. Basically, calcium is released and it helps muscles contract. So you'll see your muscle unit contract, it'll shorten, and you'll produce a little bit of tension there. Okay, so might have gotten a little bit confusing just within this last step, but we'll talk about it in just a little bit more detail now. So this little area, this step is probably quite complicated. So it actually expands, and there's a lot of things which happen before we can get muscle activation here. So let's talk, and you can find the notes again in the slide notes. What is troponin? Troponin, right here, troponin, you can see it here, and you can also see it in this diagram over, oh, you can't, no, yeah, so troponin will be along here. But what is troponin? Troponin is a regulatory protein, and it binds to actin. So it binds to this green actin strand, right? So troponin binds to the green actin strand. It is actually also bound to another protein called tropomyosin. So I'll just type that out, tropomyosin. And that's another regulatory protein. So troponin and tropomyosin can be bound together, and troponin can also bind to calcium. So 3 different things troponin can bind to. You've got the actin, tropomyosin, and calcium. Now, let's talk about tropomyosin. Tropomyosin is a regulatory protein, and it covers the myosin binding sites. So what happens is it covers myosin binding sites on the actin. So it's actually probably this one right here. It prevents myosin from being able to bind to the actin, okay? So keep that in mind. Tropomyosin prevents myosin being able to bind to actin. So first step, calcium is released into the sarcoplasmic reticulum. Calcium binds to troponin, yeah? It binds to troponin. What does that mean? That allows tropomyosin to slide away from the myosin binding site on the actin. So tropomyosin slides away from the myosin binding sites on actin, okay? What does this mean? Myosin head is now able to bind to the actin filament, like we're seeing right here. And what it does is it pulls the actin strand towards the M line. It pulls it towards the middle, okay? Or to the center of the sarcomere. This causes, so you see that power stroke, that pulling, right? And that shortens the sarcomere. And this is why this muscle activation theory is called the sliding filament theory. You see actin filament and the myosin filament, and you see the actin filament sliding over, pulling to the, in this case, towards the M line, or shortening to the center of the sarcomere. So the filaments slide over each other. However, you realize these myosin heads, they can only move a short distance before they need to be recocked, which means we have to send them back, right? And this is done by ATP, which allows them to continue pulling. So again, so far, what have we got? Calcium is released. It binds to troponin. This kind of removes tropomyosin from the picture, and that allows myosin, so the purple, to bind to the actin, the green, and it forms what we call a cross bridge. So let me just erase that here and circle this keyword. It forms a myosin-actin cross bridge, which is the idea that myosin filament can bind to the actin filament. The thick filament binds to the thin filament. Power stroke occurs, which means the myosin head pulls the actin filament towards the center of the sarcomere, so towards the M line, and it shortens the sarcomere. What happens after that? You've got the attached ADP and phosphate group are released. So this, which was here right here, is released, okay? Get rid of it. The myosin head then hydrolyzes ATP to ADP and phosphate, which means we return the cocking of the myosin head, okay? Which means we are now able, after this step, so after this ATP investment, so you're breaking down ATP to ADP and PI, you get the recocking of the myosin head, and you can do the same thing again, okay? Alrighties, so I hope that was okay. I'll run through it just in one sec. Next thing I'm going to do though is just show you the difference between the sarcomere when it's relaxed versus contracted. When it's relaxed, you can see there's a difference between the M line and the actin filaments, the myosin filaments. So the actin has not yet been pulled towards the M line. So the H area is larger, okay? The H zone is larger, or it's distinguishable, as in there's gap there. What happens when we contract is the myosin filaments, these heads, or these tiny little heads here, they grab onto the actin and they pull into the center, pull it towards the center of the sarcomere on both sides, which shortens the sarcomere. You're shortening the H zone, and so these actin filaments can almost line up along that M line. Basically, you push in your sarcomere, okay? So that's going to be the difference of the appearance of sarcomere when it's relaxed versus contracted. Okay. I don't want to be repetitive, but I know it can be really quite confusing. I remember being really confused in this class, so I'm going to go through it just one last time, keeping it nice and simple as best I can. Action potential arrives at the neuromuscular junction from the nerve. Diffuses, binds to receptors, you're starting another action potential again. This is propagated or spread along the sarcoplasmic reticulum, basically causing calcium to be released. Calcium binds to troponin. That means myosin head is now able to bind to actin. It binds to the actin, and during a power stroke, it pulls and towards the center of the sarcomere. So we're shortening the sarcomere. We release the ADP and PI. We get a new ATP formed, so coming in and binding, we break that ATP to release energy to get more ADP and PI. So we hydrolyze so that we can recock the myosin head so it can bind again and do it all again, okay? Within this process, you can see the difference between the relaxed versus contracted sarcomere. When it's relaxed, you see the difference. You see that space in the H zone here. When it's contracted, you see the shortening of the sarcomere and the disappearance or the reduction in size of the H zone, because we're pulling in from both sides, contracting or shortening that sarcomere unit. When you have a lot of sarcomeres contract, so you've got a lot of myofibrils contract, and you have muscle fibers contract. And when you have a lot of muscle fibers contract, then you can contract an entire muscle, okay? That's muscle activation for you guys. I really recommend going over this, having a read through these, search it up on YouTube if you want, having a look, see if there's any explanations there. And I've got notes in the description box. So if you have a look at the slides, there's notes there as well, going over the main ideas. But key things to know: relax and contract. Calcium binding to troponin. That allows myosin to bind to the actin filament. We shorten the sarcomere, causing that muscle contraction. And then it's a cycle, so once you get to here, you're ready to redo it all again, starting up from the top, okay? All right, let's keep moving. Muscle activation, same thing, just having a look at a slide, and it's asking you what type of muscle it could be, so probably a skeletal muscle cell. You've got the peripheral nuclei. You don't see the branching in the same way that you'd see cardiac. Cardiac muscle, there's no intercalated discs here. So we're thinking skeletal. Now it's asking us to identify and label the following components. So show them what a sarcomere is. Remember, what is a sarcomere defined by? The distance between a Z line and the next Z line, okay? So the attachment site for actin, distance between two Z lines. What is a Z line? What does it look like? You guys give this a go. There's I band, A band, H band, M line. You can hop back in the next slide, or you can pause right now if you want to try answer this. I've got the answers on the next slide. And it asks you, how would these components appear if the muscle is contracted compared to when it's lengthened, and what kind of changes you would see. So if you want a second to give this answer a go, by all means, go for it. The answer is just on this next slide. Key thing to remember here, go back to the diagram. What is a sarcomere? Distance between the Z lines. It's skeletal because it's long and cylindrical. It's got those striations. You can see that nice organization of the actin and myosin filaments in the striation. Look through this diagram to have a look at what the Z line would be, what the M line, the H zone would be. So the H zone would probably be like this dark gray area. And what would happen if the muscle is contracted compared to lengthened? When the sarcomere is contracted, the distance between the two Z lines is reduced. You shouldn't really see a very visible H zone because the actin filaments are sliding in. So again, the idea is that, you should also see the reduction in the light A bands as well. So you see the Z line move straight in towards this H zone. But yeah, have a look at that. Have a look through, kind of familiarize yourself with it. You don't have to obsess over it too much. But that takes us to the end of our little muscle part. The next thing I do want to talk about is vasculature though. Now within vasculature, they do teach you a decent variety of the tubes and whatnot, but I just want to tell you guys the main points first. So key points: all vessels have endothelial lining. So if you remember from week 5, week 6, when we talked about endothelium, we talked about how epithelium faces the outside, like our skin surfaces, right? While endothelium faces the inside surfaces. So we're talking like blood vessels and all the tubing. So all vessels have an endothelial lining, just like you can see here. You can see that endothelium right there. Vessel thickness varies due to the thickness of the tunica media and amount of smooth muscle. Yeah, so vessels vary in thickness based on their function, and that thickness should be seen in the tunica media, where you'll see smooth muscle cells, and it's related with their function. So for example, if there was a lot of blood pressure, you'd need thicker walls. So for example, arteries have thicker walls relative to their lumen size. So lumen is just the gap or the size of the hole in the middle compared to the wall size. In terms of the layers, you got endothelium, then you got the tunica intima, so this nice middle layer with the internal elastic lamina, tunica media, and then externally you got the tunica adventitia, which is where you'll find the connective tissue. I wouldn't obsess over it too much. I just really focus on the fact that thickness variation occurs in that tunica media. That's where I'd see a lot of smooth muscle, etc. Okay, next, you've got vessel wall thickness. The next thing we want to have a talk about is vessel wall elasticity. How stretchy is it? And that is dependent on the amount of elastin it has. So arteries will have a lot of elastin. Elastin is like nice and squiggly, like comes in lines and whatnot. Layers of squiggly lines. You'll notice in large vessels, you'll find more elastin in the tunica media. Again, that's where you're going to find that thickness variation. It's going to be in the tunica media, and you'll find elastin there. And lastly, low pressure veins and lymphatic vessels, which also are really low pressure. They're part of the lymphatic system, so collecting lymph, leaked fluids. They, not all of them, but many will have valves, and the valves are there to prevent backflow. I really recommend you guys having a look at these images. I'll kind of run you through the main things, but really pay attention to, I guess. Let me change color. The layers. You got tunica intima, tunica media, which is really quite thick. You can see in this artery here, which makes sense. A lot of arteries have thicker walls. And that tunica adventitia right there. In this vein, you can kind of see what the valve looks like, which I thought was quite interesting. There's an arteriole there for you to look at. Again, tunica intima, Media, adventitia. Remember, there's an endothelial lining. You've got some collagen fibers here and some smooth muscle as well. Also, just another note, vessel innervation is mainly sympathetic. There's a little bit of parasympathetic as well, but vessel innervation is sympathetic. So it's sympathetic nerves which control whether the smooth muscles around your blood vessels will contract or relax, causing blood vessel dilation and constriction. Okay? Those are the key points, the main things I really want you guys to be aware of. On the next slide, though, has a little bit of extras. Goes into a little bit of the details about the vasculature, some of their features and their functions. So, yeah, a heart pumps blood, cardiac muscle, connective tissue. You've got an artery, thick walls. You've got layers of elastin and smooth muscle. Why? Because there's a lot of blood flow. There's high blood pressure for arteries. Arterioles, not as thick, 1 to 2 layers of smooth muscle, and they also have elastic laminae. They're there because they actually provide resistance and they control blood pressure. You've got capillaries, really, really thin basement membrane, and you've got like 1 cell thick walls, really tiny, and they're really responsible for substance and gas exchange at the lungs. Lymphatic veins, lymphatic capillaries, so they might have valves, and you'll see about their role. Yeah, they have gaps which allow them to collect fluid. Veins, thin walled as well. Valves, smooth muscle with developed adventitia, low blood pressure flow. Same thing with the venule. So I won't go into that much detail here. It's just kind of for completion's sake if you guys want to have a look through it. And the last thing I'm going to do with you guys is have a look at just some examples of histology. So this is taken from an active learning, and it's just nice and annotated, having a look at vasculature. So you guys can spend more time having a look at it later, but the main things I want to show you, you've got that internal elastic laminae, black line here. You've got your tunica intima along here as well, tunica media, tunica adventitia, really quite developed here. What is going to be in each layer? Tunica intima will have endothelial cells, there's connective tissue. Media has smooth muscle cells, varying amounts of elastin or collagen or proteoglycans, depending on the vessel and its role. And then you've got the tunica adventitia, so that will have connective tissue with elastic fibers. You can have a look through this image a little bit longer as well later if you guys want. Got one last one as well. Another one, you can see there's an artery here, lots of red blood cells inside it. You can see that lovely layer of thick tunica media. You can see a vein here, so its tunica media is nowhere near as thick. Venule even smaller than the vein, and you can even see tiny little capillaries, tiny walls, like a red blood cell or two going through those. Okay, so that wraps up vasculature histology. Again, if you have questions, let us know. Otherwise, that's it from me.
All right, so welcome back to PS Pain Relief. Thank you guys once again for all your patience. We really appreciate everyone who even just tunes in for these videos, for everyone who clicks on the PowerPoints. So yeah, just me getting a bit touchy-feely, no big deal. So my topic today is going to be blood. And what are we covering under blood? So we're covering the components of blood, haemopoiesis, blood cells, including erythrocytes and leukocytes, and we're also covering laboratory testing and blood disorders. And I will not be covering blood vessels because you guys have literally just heard it like 5 seconds ago. So let's get into it. Okay, so what is blood made of? Blood is made of liquid elements and formed elements. So under liquids, we have plasma. Plasma accounts for 55% of blood. Plasma is basically just the fluid medium that everything is dissolved in, from nutrients to metabolites and proteins. It's basically water-based, remembering that. Okay. And blood is also comprised of erythrocytes and the buffy coat. So these are the formed elements. So the erythrocytes, they account for 45% of the of the blood in total. And erythrocytes basically refer to red blood cells, and these are going to be what transports your oxygen to your tissue cells. And for buffy coat, we have
less than 1% of blood being the actual buffy coat. This buffy coat refers to leukocytes and platelets. Leukocytes are white blood cells. They're the basis for the immune system, and platelets enable the clotting of blood. Okay, so yeah, not super difficult. You guys probably have a handle on this already. And so now haemopoiesis. Haemopoiesis is basically how hematopoietic stem cells are formed into blood cells. And as you guys can see here, I have provided this handy dandy diagram. So you guys can see how multipotent hematopoietic stem cells can become either myeloid progenitors or lymphoid progenitors. And remember that your erythrocytes come from myeloid progenitors, and your basically your adaptive immune system comes from lymphoid progenitors. Okay, that's my messages going off. Okay, and also we have thrombocytes. Thrombocytes are basically platelets. They're just a different interchangeable name for platelets, so don't get too caught up with the etymology of everything. And we have our basophils, neutrophils, eosinophils, and macrophages also coming from myeloid cells. So haemopoiesis, this is not super important, but basically when you're in the prenatal stage of life, it happens in the yolk sac early on, and then the liver, and then the bone marrow, when you're just about to pop out of the womb. And then once you reach postnatal development, so basically everything from once you're outside of the womb, it happens in your bone marrow, most commonly your vertebra, and also your pelvis. And there are some other areas, but once again, all you guys really need to know is that it's a bone marrow thing, okay? And so now also we need some specific minerals to be able to have haemopoiesis happen. So these are going to be iron, vitamin B12, and folate. So iron is absorbed in the duodenum, vitamin B12 is absorbed in the jejunum, and folate is absorbed in the ileum. And these three, I don't expect any of you guys to actually know what this is, but these three are basically parts of the small intestine. So you guys are going to learn in year 2 that different parts of the small intestine kind of prioritize the absorption of different minerals. So the duodenum is the first part, the jejunum is the second part, and the ileum is the third part. Yeah, so just bear that in mind, that the body finds it so important that we have these 3 minerals, that it has literally dedicated parts of the intestine to absorb this stuff, amongst other nutrients. And okay, so blood cells, very quickly, we have erythrocytes, which you guys already know are red blood cells. They have a biconcave appearance, and they have no nuclei, hence the biconcave appearance. They have hemoglobin, which are basically 4 globin chains, so 2 alpha and 2 beta chain. What am I doing with my hands? So 2 alpha and then 2 beta chains, and then they also have the heme group, which is primarily iron and biliverdin, which I've just put down here. Again, that stuff is very, very, very much year 2, but I just thought that it would be cool if you guys have the whole picture. Then you can, then it can what am I saying today? Then it kind of links up a little better in your brain. So erythrocytes are also primarily specialized for gas transport, and they're synthesized via erythropoiesis, which is mediated by erythropoietin. Once again, do not need to know that. That is very unimportant. And when erythrocytes die and they're broken down, mostly in the liver, heme and globin get separated from hemoglobin. So globin is basically recycled. Heme is broken down into iron and biliverdin. Iron is recycled once again, and biliverdin becomes bilirubin, which becomes stercobilin or urobilinogen. So verdin means green, and then bilirubin means like, I think yellow or brown or something like that. Don't quote me on that. And then it becomes stercobilin, which colors our brown stool, and it can also become urobilinogen, which colors our yellow urine. And fun fact, bilirubin is also the chemical in charge of that, not really in charge, but the chemical responsible for making people with liver failure and blood disorders look very jaundiced. So that's when your skin takes on a very yellow appearance, because bilirubin is yellow and it can accumulate in your bloodstream and just make you look really yellow. It's ironic that I say this as an Asian person, but it gets yellower than that. Sorry. Okay, leukocytes. Leukocytes are highly variable. There are many shapes and sizes. They include eosinophils, neutrophils, basophils, monocytes, lymphocytes, and plasma cells. Say that really fast 3 times. I dare you. And essentially, leukocytes may have lobed nuclei, which give them the appearance of having multiple nuclei. But remember that they're merely lobed. And their primary function is the immune response, as you guys have learned from the adaptive immune system last week. That's what they do. They are produced via leukopoiesis, which is mediated by interleukins, colony-stimulating factors, and immunological stimuli. So these are very responsive to the war that our body is engaging in against pathogens, basically. And then for laboratory testing, when you order a blood test, you usually say, I want a full blood count, or I want a full blood panel, because you'll get the numbers of all of these. There are normal values, but don't really bother remembering them, because lab reports are going to tell you maybe with an asterisk or with some bold text or a different colored font, which values are abnormal, or they're going to provide the normal range so that you can compare the test values to the normal range. So don't really bother remembering any of that. Just know that this is very, very useful in trying to detect whether or not you have an infection, because once you have an active infection, you also know that your immune system has been called down to deal with that, and so your total white cell count would be much higher than in a normal person. So yeah, just remember that. And peripheral blood film. This basically enables you to visualize the shapes and sizes of your different blood cells. So I'm so sorry. So basically, if you have sickle cell anemia, you would probably be able to see that on the peripheral blood film, because you would be able to see the shapes and sizes of your blood cells. And last but not least, this part is really not super important. Just know that there are a whole number of things that can go wrong with your blood, such as the number of blood cells, which again can be detected in this full blood count panel. You could detect abnormalities in the number of cells, morphology of the cells, basically the shape and the size, which can be detected in the peripheral blood film. You can have the functionality of the cells also being kind of abnormal. You can have the membranes being a bit off also. You can have hemoglobin dysfunctions. Remember that when you have carbon monoxide poisoning, fun fact is that your red blood cells prefer to bind, or more specifically, your hemoglobin prefers to bind to your to carbon monoxide over binding to oxygen. So I mean, that's kind of, if you ask me, that's kind of like a design flaw in the human body, but what are you gonna do? Just don't inhale carbon monoxide. And then we can also have metabolism going wrong. So this specifically refers to the metabolism carried out by blood cells, because obviously they also need energy to function properly and to be able to form a working immune system or a working oxygen transport system. So that's literally all you guys have to know for this bit. It's not a lot of stuff, and thanks for staying with me till now, and on to the next person who is going to talk about cancer. See ya.
Okay, moving on to a bit of a heavier topic now. I'm going to talk about the molecular basis of cancer and how it affects the patient. So starting off, I'm going to talk about the different types of cancer. So obviously different types of cells will give different types of cancers. And the most common type of cancer are your carcinomas. And these arise from skin and epithelial cells of organs. They account for around 85% of cancers, and they include breast cancer, lung cancer, colon cancers. Your sarcomas are much more rare. They arise from bone, muscles, and blood vessels. And finally, your lymphomas, they arise from your lymphoid cells. So think your lymphatic system. And leukemias are specifically from white blood cells of the bone marrow. So some characteristics and behavior of cancer cells. I've included this little table here. So basically, this will kind of help you in the histological section. So when you look at cancer cells, they will have a larger nucleus and a relatively small cytoplasm compared to your normal cells. And the nucleus will also be variably shaped, so it won't have a regular shape. You'll also have variation in cell size and shape due to the changes in the cytoskeleton. So it will have a different shape to the cells next to it. And there'll also be a disorganized arrangement of cells. Cancer cells will also have a loss of normal specialized features and an elevated expression of certain cell markers. And there'll also be a large number of dividing cells and poorly defined tumor boundaries. So unlike normal cells, cancer cells don't need to attach to a physical substrate to grow, meaning that they can grow in clumps. So they can kind of grow on top of each other and form clumps, whereas normal cells would have to grow in a monolayer. And because normal cells grow in one layer, they're also in close contact with each other, which also inhibits cell growth. And that's why cancer cells also proliferate so much, because they don't need to grow in one layer. They can just grow in clumps, and so they don't have anything that inhibits it from growing more. Also, because they grow in clumps, they have the ability to move around, unlike normal cells, because they don't attach to the surface that they're on. So cancer cells have a greatly reduced requirement for growth factors. So they don't need as many growth factors to grow, leading to a faster/higher rate of proliferation than normal cells. Cancer cells are immortal, and they don't stop dividing after a certain number of division cycles. So this means that they can accumulate more mutations, and telomeres play a key role in this. Cancer cells also resist growth inhibitory signals, which leads to uncontrolled growth, and they have many chromosomal abnormalities, including changes in chromosome number and structure. Okay, so let's talk about the telomeres. So the shortening of telomeres equals an aging cell. Okay, so normal cells will undergo more DNA replication, and their telomeres will also shorten as a result of this. And short telomeres are also a mechanism of inhibiting cell growth. So old cells that have super short telomeres will eventually stop growing, and this is what we want. However, cancer cells can re-expand the telomeres by re-expressing telomerase, and this tricks the body into thinking that it's healthy despite being damaged, and then it's able to continually grow and mutate. So when we talk about tumors, you can either have a benign tumor or a malignant tumor. So the progression of cancer can actually be described as going from a single mutant cell to forming a tumor to metastasizing. So that's just spreading around the body. And tumors are masses of cells derived from a single cell and can be benign or malignant. And benign tumors can become malignant as well. So I've included this table that outlines the differences between benign and malignant tumors. So benign tumors are called adenomas. They represent a low level of genetic damage. They'll resemble the cell or tissue of origin. So they'll actually be what you call identifiable, because you can identify where it came from. Benign tumors also remain localized and don't spread. And they don't usually cause problems unless they grow in a confined space, such as the brain. Malignant tumors are what we call adenocarcinomas. They represent a high level of genetic damage, and they're usually unidentifiable, so they don't resemble the cell or tissue of origin because they've mutated so much that they've developed irregular structures, large nucleus, little cytoplasm, etc., etc. Malignant tumours will invade surrounding tissue and cause many problems, so they will spread. You can see just an image here, the difference between benign and malignant. Okay, so angiogenesis and tumour growth. So angiogenesis is the formation of new blood vessels due to growth factors. So blood supply is important for cell growth as it supplies oxygen, nutrients, and removes waste products. Tumours secrete a growth factor called vascular endothelial growth factor, VEGF, which induces angiogenesis and forms new vasculature around the tumour. So the tumour can actually, the tumour basically can like almost essentially control blood growth around it and basically tell the body to feed it so that it can grow. And angiogenesis is necessary for benign tumours to become potentially metastatic. Okay, so moving on to tumour metastasis. So usually you'll start with a benign tumour in wherever. So in this example, you have in your epithelium. It will grow and grow and grow. Eventually, it will break through the basal lamina. Also, it will grow due to angiogenesis. So there's a blood supply feeding it, nurturing it, nourishing it. It'll grow so big that it eventually breaks through the basal lamina. And then from there, it can invade the capillaries. It can travel throughout the body. And then it can either adhere to capillary walls. It can escape from capillaries. That's what we call extravasation. And it can even proliferate to form metastasis wherever it goes. Okay, so this is just in the liver, for example, but it can go other places as well. Okay, so the way that it breaks through the basal lamina is because the cancer cells can release proteolytic enzymes, which is just enzymes that break up things. Local spread means that the tumour has metastasized to nearby tissue, and distant spread is it's entered the bloodstream and metastasized to lymph nodes, distant tissues, and organs. Okay, so DNA damage is what causes cancer. So cancer is caused by the accumulation of genetic mutations in cells. So it's not just, usually not just one mutation that will lead to cancer, it's the accumulation of many different genetic mutations and other factors such as the age of the cell, the fact that it can't function anymore, and all of this will result in increased cell growth and resistance to apoptosis, proliferation and invasion of other cells, and the ability to evade the body's immune system. So DNA damage can be inherited, which is what we call germline errors. And this is when DNA mutations are inherited from a parent's egg or sperm cell. So every cell in the resulting person will contain a copy of the damaged DNA. So this is present throughout their life, and it makes up 10% of cancers. DNA damage can also be acquired, so we call that somatic errors. And it's when DNA mutations occur at some point in an individual's life and will be present only in certain somatic cells. So it can't be passed down to the next generation. And it's caused by things like exposure to UV light, smoking, radiation, viruses, etc. And it can also be caused by errors in DNA replication. Of course, we usually have mechanisms to repair DNA damage, but too many errors or loss of DNA repair enzymes or less time for DNA repair due to rapid cell division will lead to an accumulation of DNA damage and mutations. Cancer cells also have a more rapid cell cycle, so there's faster accumulation of genetic damage. So basically, just keep in mind that it's a lot of different factors that lead to DNA damage that eventually then lead to cancer. Okay, so oncogenes in cancer development. So oncogenes are mutated forms of proto-oncogenes that lead to increased and inappropriate cell growth and division, which predispose to cancer. So proto-oncogenes are just normal genes that promote cell growth and division, okay? But your oncogenes are what you call proto-oncogenes that have mutated, and therefore you have increased cell growth, therefore predisposition to cancer. Oncogenes act in a dominant manner. So the gain of function in a single copy of a cancer-critical gene can predispose to cancer. It follows what we call the one-hit hypothesis, which is what you'll need to remember, which basically just means that you only need one gene to be mutated to cause a cancer, okay? So you can see here, normal cell has 2 genes, and then you'll have a single mutation event that creates an oncogene, and it only needs to occur in one gene, in which case you may have cancer. So an example of an oncogene, when we look at something called chronic myeloid leukemia. So chronic myeloid leukemia originates from a single abnormal hematopoietic stem cell. It's characterized by unchecked proliferation of immature white blood cells in bone marrow and peripheral circulation that leads to bone marrow being replaced by immature cells, an increase in liver size, and a massive increase in white cells in the circulation. So CML is caused by a translocation between the long arm of chromosome 9 and the long arm of chromosome 22. So these two, the long arm of these two chromosomes are essentially swapped. Now, on chromosome 9, we have what we call the ABL tyrosine kinase gene, okay? And on chromosome 22, we have what's called the BCR gene. So when you have a translocation of these, between these two chromosomes, you'll get a fusion of these two genes, and it will create a fusion protein that leads to increased tyrosine kinase than normal, and this is what causes leukemia. Okay, so tyrosine kinase receptors are just the receptors that respond to extracellular growth factors, and therefore they regulate cell growth. Okay, so when tyrosine kinase receptors are mutated or overexposed, this causes overexpressed. Sorry, this is what causes cancers. Okay, this is what we call. This is an example of oncogenes. Okay, because you have overexpression leading to cancer. Another thing that can relate to cancer development is your tumour suppressor genes. So your tumour suppressor genes are normal genes that are present in all cells that act to prevent cancer by inhibiting growth. So you kind of have a balance between your tumour suppressor genes and your proto-oncogenes, right? A balance between promoting growth and inhibiting growth. So tumour suppressor genes code for things like cell cycle proteins, transcription factors, cell surface proteins, DNA repair proteins, and apoptosis-related proteins. They control critical points in the cell cycle and induce transcription of regulatory inhibitory genes. And they also encode proteins that inhibit, so basically they encode proteins that inhibit cell growth. Now, in oncogenes, we saw that it was a gain of function that led to cancer. Now, in tumour suppressor genes, you need a loss of function in the gene to predispose to cancer. And you need the loss of function of both tumours, both tumour suppressor genes, sorry. So therefore, you follow the two-hit hypothesis, where both genes need to be mutated to cause cancer. If one gene is inactivated, there's only a slight growth advantage in the cell, but it's not really enough to cause cancer. You really need both genes being mutated. So an example of this is the retinoblastoma gene, what we call RB. So the loss of function in the tumour suppressor retinoblastoma gene leads to retinoblastoma, which is a rare childhood tumour of the retina. There's two forms of the disease. It can either be hereditary, which develops at a very young age. The common sign of this is having tumours in both eyes. and and you'll see the mutant RB allele on chromosome 13 in every cell of the body. Okay, remember, because it's hereditary, it's passed on, and the individual will have the mutation in every cell of the body. The other form is sporadic, which is non-hereditary. It develops at a later age, and its characteristic is only one tumour in one eye, and the chromosomal changes will only be present in the tumour cells. The RB gene is also mutated in other cancers, including breast, lung, and bladder cancer, alongside other genetic mutations as well. Another example of tumour suppressor genes in cancer development is the tumour suppressor p53 gene. So mutations in the p53 genes are one of the most common mutations. So 50% of all tumours will show mutations and loss of function of p53. So let's take a step back. In biology, we learned about apoptosis, which is a normal controlled process that regulates cell death, and it's a good thing. You generally want apoptosis to occur. Now, p53 gene functions include inducing cell cycle arrest, senescence, cell differentiation, and apoptosis. P53 gene also contributes to DNA repair by after damage, or if damage is severe, it will induce apoptosis. So essentially, its main role is to inhibit cell growth when it's inappropriate, thus preventing tumor development. Your mutations in p53 genes will help cancer cells evade apoptosis, thus promoting proliferation. It will also lead to DNA damage not being repaired, and the survival of mutated cells. And a common result of a mutated p53 gene is that chromosomes will become fragmented, they will incorrectly rejoin, and you will have an increasingly mutated genome as cell division continues. Moving on to mismatch repair genes. So MMR genes repair DNA damage during DNA replication, and they're often lost or defective in cancers. Replication areas are seen in hereditary and sporadic cancers, including colorectal, endometrial, breast, prostate, lung, and stomach cancers. Hereditary non-polyposis colorectal cancer is due to a loss of mismatch repair genes. They also follow a two-hit hypothesis. That should say deficient, so I'll fix that up. So two-hit hypothesis leading to cells that are deficient in the mismatch repair genes, and in total, humans have 8 MMR genes. So let's start talking about the effects of cancer. So cancer can have both local and distant effects. So your local effects include tumours displacing surrounding tissue, and the function of adjacent tissue deteriorating as normal cells become cancerous and unable to carry out their specialized functions. So, for example, you have leukemias replacing normal bone marrow, and therefore blood cells can no longer be made, or brain tumours replacing normal brain tissue, and you won't be able to carry out certain body functions anymore. And also tumours can become so large that they block vital passages. So local colon tumours can block the lumen of the bowel, and tumours can also block arterial, venous, or lymphatic flow. Now, your distant effects are just anything that can occur when the cancer spreads. So, for example, if the cancer spreads to the bones, you can have pain and fractures. If it spreads to the brain, you can have headaches, seizures, or dizziness. If it spreads to the lungs, you can have shortness of breath. If it spreads to the liver, you can have jaundice or swelling in the belly. And again, it can also block vital passages. Why are cancer patients often cachectic? So this is something you'll learn maybe next semester or this year when you have an older age patient coming in and they're obviously cachectic. So cachexia is when a patient's very underweight. You generally want to think straight away, could could this person have cancer? And this is because cancer cells have a rapid growth and turnover rate, leading to a high metabolic rate in the individual. Cancer cells also compete with normal cells for nutrition and blood supply, and vital organs may break down their own proteins to nourish the cancer. So this is why often many cancer patients present with cachexia. Cancer dormancy is also another important concept. So breast cancers and melanomas often express dormancy. And this means that the cancer can reappear decades after the initial tumour has been removed from the body, even if there's been no evidence of cancer in those years. And dormancy may be related to an increased acquisition of blood supply, leading to tumour growth once again. So that's your angiogenesis. Okay, so let's talk about the features of inherited cancer predisposition. So I said earlier that about 10% of all cancers appear to run in families. The consequences of this is that you need to have routine screening for cancers, such as breast and bowel cancer, in susceptible families. Families may also require genetic counselling to understand the implication of genetic predisposition. So this is important to know what the features of inherited cancer susceptibility syndrome are. So how can you tell if someone may be susceptible to cancer? Okay, so these are the features. Either they have one or more of these dot points. Okay, so first of all, if they have several close or first degree relatives, so that's your mom, your dad, your siblings with a common cancer, that's with a common cancer. Okay, so common cancers are like breast cancer. Or if you have several close or first degree relatives with related cancers. So this is breast cancer and ovary cancer. If someone has two members of the same family with the same rare cancer. Okay, so if you have an auntie and a cousin with the same really rare cancer, then that could be a feature of inherited cancer susceptibility syndrome. If there's early age onset of cancer, that's another feature. If there's bilateral cancers in paired organs, so for example, cancers in both kidneys, that could be a sign of predisposition as well. Or if there are tumours in two different organs in one individual. Now, when trying to identify if a patient has a genetic predisposition, you have to still take into account the following. So some cancers may just be very common, such as breast cancer, where 1 in women will get it, okay? So some cancers may just be very common. It might not mean that there's a familial predisposition for it. And also, depending on the gene, not everyone who has a specific genetic defect will get the disease associated with it. And this is what we call penetrance. So some colon cancer syndromes have a penetrance rate of 100%, while inherited breast cancer syndromes have a penetrance rate of 40 to 70%. Let's hone in on breast cancer a bit. So up to 10% of breast cancer is due to genetic predisposition. And the hereditary breast cancer genes include BRCA1 and BRCA2. Other cancers are also attributed with this inherited mutation, and it includes ovarian cancer, colon cancer, and prostatic cancer. And some features that indicate an increased likelihood of someone having BRCA mutations includes multiple cases of early onset breast cancer in the family, ovarian cancer with a family history of breast or ovarian cancer, breast and ovarian cancer in the same woman, bilateral breast cancer, Ashkenazi Jewish heritage, and male breast cancer. Now, if you know that a patient is at risk for familial cancer syndrome, prevention or early detection is key. So you run screening tests, okay, and gene testing to look for early evidence of the clinical features, so things like colonic polyps or breast lumps. But you'll also need to weigh the benefits of screening long-term versus the physical or emotional toll on the patient. And you'll get more about this in semester 2 when you learn about population health. Finally, your different cancer treatments include radiotherapy, so using radiation to destroy cancer cell DNA or kill cancer cells or stop them growing. Chemotherapy, which is using cytotoxic chemicals and drugs such as methotrexate to kill cancer cells, but this also damages our own cells, such as your hair cells. Surgery, so cutting out tumours, and monoclonal antibody therapy, which is mass producing your antibodies to target cancer cells. So this will just help your immune system to attack the cancer cells. And that's it for cancer.
Hello, Jaffies. I hope you are all doing very well. Today I'm going to be talking to you guys about pharmacology. We're going to be looking specifically at the basics of pharmacodynamics, a little bit of nomenclature of 6 common drugs that you guys will probably be talking a lot about this year. And we're also going to talk a little bit about the general modes of action of most drugs that we see today and where these guys come from. So to start with, we need to talk about general definitions of a drug. This may seem pretty straightforward, but it is very important to have these basics down pat. So there's 2 basic features to every drug. The first is that, as you've probably worked out, it's a chemical that changes the way our bodies work, and specifically it does that on a cellular level. So it changes the way our cells work. Now, the other feature of drugs, in order to be a drug, is that they have to not be a natural component of our diet. So in other words, the chemicals in the red meat that we eat are not considered drugs, or the chemicals, all the vitamins that we find in fruit and veg, these are not drugs, even though they do change the way our cells work. So based on that definition, see if you can work out which of these guys would be considered drugs. So you can feel free to put your video on pause and just have a little think if you need, and we'll move right on. So in other words, all these circled ones here, we would call these drugs, while the non-circled ones are not considered drugs. And the reason for that is that the circled ones don't exist naturally in our diet, and they do satisfy the first criteria, which is they change the way our cells work. The other ones, so B, D, and E, these all naturally exist in the diet. It's just that they've been concentrated into these, what we call dietary supplements here. So here we got an EpiPen, we got Panadol Osteo. This one here is what we call a cyclooxygenase inhibitor. And then finally, we got a calcium channel blocker here, diltiazem. So the Panadol and the diltiazem have a little something in common, and that is that they don't exist in the body. Whereas EpiPens, well, EpiPens themselves obviously don't, but they contain adrenaline. And adrenaline is a neurohormone and a neurotransmitter. So it naturally exists in the body. So as an extra classification, we would call an endogenous drug. All right, so here's some basic nomenclature summarized in a convenient table. We all know med kids love tables. So I've put the first 4 in bold here, and you'll see that that's because they have similar function, and you'll hear about these guys over and over and over again. They're probably some of the most abundant medications. We'll start with beta blockers. Olol is probably the most common suffix you will see on medical charts. It's everywhere. So these guys are beta blockers, so specifically beta adrenoceptor blockers, and their function is to reduce blood pressure. Same with ACE inhibitors and these calcium channel blockers. Now, it's worth noting that the dipines, we're talking about what we call dihydropyridine calcium channel blockers. You'll learn more about the specifics of how these are different to what we call non-dihydropyridine calcium channel blockers. But the easy way to remember that they are dihydropyridine, because it is clinically relevant, is a dipene dihydropyridine. So they kind of sound similar. And then we've got these statins here. So these don't reduce blood pressure, but they do reduce blood cholesterol. So they've got a similar sort of cardiovascular role with these other 3 drugs here. Things with a cane ending, so we're thinking lignocaine, and this numbs the skin. It's a topical or local anesthetic, so it numbs the skin or the mouth. And then we've got tadines. So we're talking about histamine H2 blockers here. And what these do is they're good for what we call gastroesophageal reflux. So you take them and then they block histamine receptors in your stomach, and that stops histamine from causing those cells to release hydrochloric acid. So you reduce acid secretion, you can help to combat GORD. Worth noting, statins, you might remember from my little talk to you guys on lipid metabolism, that HMG-CoA reductase is a key enzyme in the synthesis of cholesterol. And so by blocking that, by inhibiting it, we reduce cholesterol, and that's the function of the statin. All right, so now let's move straight on to pharmacodynamics. We're not really going to talk too much about pharmacokinetics, but it's important to understand its distinction with pharmacodynamics. In short, pharmacokinetics is talking about what the body does to the drug, so it absorbs it, it distributes it throughout the body, so usually via the blood or the vasculature. Then we metabolize it, so we process it, break it down into smaller bits called metabolites, and then finally the body excretes it. Pharmacodynamics is more talking about it from the drug's perspective. So we can remember pharmacodynamics with the D is talking about what the drug does to the body. And in pharmacodynamics, almost all drugs do fundamentally the same thing, and that is that they bind to proteins, but once they bind to proteins to cause their effect, they then have differences in the way they operate. So one way they can work is that they can activate that protein. They can cause that protein to do some bodily function, or they can not activate the protein, and in doing so, they block things that would activate the protein. So when we activate it, we call this agonism, and when we don't activate and we block it, we call this antagonism. And then, of course, we're talking about what is the effect of this agonism or antagonism in terms of what the target protein and within the cell then does. Little example for you down the bottom. So here we've got a little summary of how that works, complete with a diagram. So first, the drug will come along. It's here in this little red oval here. It will bind to the target protein. Almost all drugs work by binding to proteins. This is almost universal. Once it's bound to the protein, then it can do one of two things. It either changes the protein shape, changes the molecule shape, or it doesn't. Now there's actually a few versions of this story, but we're going to use this version because this is relevant to what we call receptors. And we'll talk about why it's relevant to receptors as opposed to other things shortly. Anyway, we change the molecule shape. Now let's say that the changed form is the active form. So if we change its shape, then that would be an agonist. So it's activated the protein. Now, on the other hand, if we look at this middle one here, we can see that the protein shape has not been changed even though the drug has bound to it. So currently the drug is acting as an antagonist. It's blocking things that would activate the protein from binding to it. So it's an antagonist here, but in the red section here, it's being an agonist. So let's talk a little bit about receptors now. These are almost always the targets for drugs. There's a few exceptions. Sometimes drugs target enzymes, in which case it kind of tends to work the other way around, and we'll talk about why that is. So in this case, we're talking about a beta adrenoceptor, or a beta adrenergic receptor as it's listed here. Epinephrine is just the American word for adrenaline. So adrenoreceptors receive adrenaline. And you can see it's got this little G protein down here. That's like a little extra protein. Now, if this main receptor here gets activated, it's going to change shape, which mobilizes this G protein, which activates the G protein, and then that G protein is going to activate the next thing, which activates the next thing, which activates the next thing. We call this type of reaction a cascade reaction. Also, you can kind of simply call it a chain reaction. So if a drug was to act here, let's say the drug is adrenaline, it binds to the adrenoreceptor, that changes the adrenoreceptor's shape, activates the G protein, and sets off that chain reaction. Now, let's say we have a drug that binds to the receptor but doesn't change its shape. Well, then it's not going to move the G protein. So it's not going to set off this chain reaction. We would call that an antagonist. Or because it's blocking the epinephrine from binding to this receptor, we would call it a beta blocker. So this is the function of a beta blocker. Now, this diagram is just summarizing what I was saying before. So at this point, in the center, we're talking about antagonism because it's blocking, it's not allowing the target protein to perform its function, but it can also act as an agonist. So we can also think of this in a more pharmacological sense. The pharmacological way to think about this is using these two principles of affinity and efficacy. Affinity is talking about how much the drug likes the protein and vice versa. And efficacy is talking about, well, once they're bound together, how active does the drug protein complex become? Or rather, how much time does it spend in this active state here? So let's start by looking at what high affinity means for a drug. Well, high affinity means that the drug is going to spend a lot of time bound to this protein here. So drugs with high affinity, once they meet their target protein, they move pretty much straight into this stage 2 here that you can see. Now, it is what we call an equilibrium. So in other words, what's really happening is that the drug and the target protein are moving between two states, a bound state, or what we call a complex state, and a dissociated state, which is denoted by 1 here, which is where they're separated. But when a drug has high affinity, we say that it spends most of its time, or pretty much all of its time, in this complexed state. Now, efficacy then basically tells us whether our drug is an agonist or an antagonist. So when it's active, we know that the shape has been changed. So high efficacy means that the drug is what we call an agonist. Whereas if the drug has 0 efficacy, it's going to spend all its time in stage 2 with the protein's shape not changed at all. So it's going to be inactive. What this means is that drugs with high affinity and high efficacy, they stay in stage 3 and we call them agonists. And drugs with high affinity and 0 efficacy are what we call antagonists because they stop the protein from functioning. So this is sort of the complete pharmacological story. We were looking at the basic story before. This is the complete story. Now, it's worth noting there's another class of drugs called partial agonists. And what these guys basically do is they flip and flop a bit even Steven between stages 2 and 3. So they spend a little bit of time as an agonist and a little bit of time as an antagonist. Here's a little table that summarizes this. And the takeaway that I want you guys to get from this is that when you have a drug with high affinity, that is always ideal, regardless of its efficacy. You want it to be high affinity, and then the efficacy will determine its role. So if you have a high affinity drug, that just means that you need less of it to achieve the desired effect. Whereas if you have low affinity, then you need to administer more of the drug and you have the risk of complications elsewhere in the body. So high affinity is way preferred. If you have low affinity, it might bind to something you don't want it to bind to, for instance. Now, that's not to say that you should always be using a high affinity drug because it needs to have the correct efficacy. For instance, you don't want to give beta blockers to an asthma patient. That's because beta blockers can also cause bronchoconstriction by more or less preventing bronchodilation. So we need to make sure that we're using the right thing between an antagonist and an agonist. Now, last thing I'm going to cover about this whole story is just this induced fit model. I'll keep it real brief. Basically, our drug, let's say that enzyme here is instead drug, and substrate here. Let's say enzyme is target protein, and let's say substrate is drug. Well, the target protein is a protein, so it's a big molecule and it's a little bit flexible. Now, the shape of the target protein needs to be complementary more or less to the drug. But if it's not perfectly complementary, that doesn't matter. There's a bit of wiggle room where they can still sort of fit together. They just conform to each other's shape. So that's just the induced fit model. Now, the last thing we need to talk about is where do drugs come from? And whilst it's low yield, it's still incredibly clinically relevant. So the vast majority of drugs come from nature. The most important thing to know is that some drugs come from nature. The effect of some drugs is discovered by accident. And then some drugs are what we call rationally designed. So we actually design the drugs. But this is a this is incredibly difficult to do, and it's more of a developing area of drug development. Let's talk about the main area of drug development, which is where we get drugs from nature. Almost all of the drugs that we use therapeutically in hospitals today come from nature. So we've got aspirin, for instance, one of the most abundant non-steroidal anti-inflammatory drugs. Also lowers blood pressure a little bit. And this originally came from willow bark, and it was isolated in the 1800s. We've got opioids, which give us codeine. They also give us some pretty nasty drugs like heroin, but they give us morphine and fentanyl, which are incredibly, incredibly important analgesics. Basically, without this kind of pain management drug, there's so many things that wouldn't be possible. Things like post-operative things would just be so painful, for instance. So they're very, very important. Palliative care would not exist if it weren't for drugs like this. And then penicillin. Well, this penicillin antibiotics revolutionized the world of medicine in the 20th and 21st century. Without penicillin antibiotics, we would be helpless against so many infections that present to hospitals every single day. So the takeaway from that is that biodiversity is super, super important for the future of pharmaceuticals. And because modern medicine basically hinges on good pharmaceuticals and good pharmacological management, the future of medicine really depends on good biodiversity. That's the end of my lecture. If you have any questions, feel free to ask us directly or leave them in the Google form.